Hi there, my name is Ling Qi, and today I'm going to talk about our work Bayesian image reconstruction from retinal cone signals. I would first like to thank my wonderful collaborators, Nicholas Coteras and David Brainerd. I would also like to thank our funding source that made this work possible. There is a long tradition in studying the limits of our visual system in terms of information processing using simple stimulus, such as grading, and simple psychophysical experiment, such as contrast sensitivity, measured using detection, and how it changes as a function of increasing spatial frequency. We also have pretty good understanding of experiment like these using ideal observer analysis, which detailed characterize various components of visual system and the statistical properties of the experiment. We're interested in extending this type of analysis to use natural images. This requires us to make our ideal observer more image computable. In our talk today, we will focus on components of early vision, including optics and properties of photoreceptors. We would also like our ideal observer to take into consideration the statistical regularities of the natural environment. To work with natural image, we need a task that goes beyond simple threshold measurement. Thus, we ask our ideal observer to perform image reconstruction from the signal available to our visual system. From the reconstructed image, we can perform a wide range of analysis, including threshold, and they also provide us with a direct visualization of information loss. Concretely, we take a Bayesian approach, meaning we first construct the likelihood function, which describes the probabilistic relationship between stimulus and pattern of cone excitation. We do this with the open source software EyeSightBio, which is an image computable model of early vision that I'm going to introduce to you in part 1. Next, we construct our Bayesian estimator, and finally, in the third part of the talk, I will show you some analysis of how variations in early vision influence image reconstruction. Let's start with our likelihood function. To recap, we need to model how stimulus gets translated into pattern of cone excitation. We start with showing some image on display. So the red, green, and blue channels of the image modulate the 3 primaries of the display, each generate light energy within specific wavelengths. This produces a pattern of spectral irradiance that we are visualizing here at 9 example wavelengths. We model the transmission of light through physiological optics using point spread function measured at different wavelengths. The point spread function measures the response of an imaging system to a point source. We also measure the absorption of short wavelengths by human optics. This gave us the optical image that we can also break down into different wavelengths. And as you can see here, for example, the effect of chromatic aberration at short or long wavelengths. This pattern then gets absorbed by a cone mosaic. For example, here, the red, green, and blue circle represent L, M, and S cone, respectively. And we finally get our pattern of cone excitation. The brightness here represents the degree of cone isomerization. The last component we need is a noise model, and we know cone isomerization is a Poisson process. Thus, the final form of our likelihood is the independent Poisson distribution on each cone with the rate parameter calculated by our EyeSightBio routine. With the likelihood in place, we can start to construct our Bayesian estimator. We first need a prior distribution of natural image. The basic idea is we want to capture the high-dimensional joint distribution of pixel value, especially their correlation and dependency. We do this with a simple sparse coding model. The basic idea is for any small patch of natural image, we can model it as linear combination or weighted sum of basis image. If we choose those basis image from a set of oriented edges at different scale, the distribution of coefficients can be characterized as sparse, as shown here on the right. So the red distribution is the distribution of coefficients, and the dotted black line shows a Gaussian distribution for comparison. As you can see, the red distribution is peakier at 0 and has longer tail. We extended this analysis to color image by learning a set of basis functions using independent component analysis on a large data set. These are examples of some of the basis functions. As you can see, they all look like oriented edges, but there seems to be 3 distinct classes: achromatic edges, yellow-blue edges, and red-green edges. We also verified that the distributions of coefficients are indeed sparse. So with other components, we can now start to construct our Bayesian estimator. We use the maximal A posterior estimator. The basic idea is for a pattern of coexcitation, we find our estimate, for example, as shown here, by starting with a random image and start to adjust the pixel value iteratively to maximize the product of the likelihood and prior, or the sum of the logs. And to just recap, the likelihood function is a Poisson distribution with the rate parameter calculated by isolated bioroutine. And the prior distribution, we project images onto a set of basis function and assert the distribution of coefficients are sparse by, for example, using a Laplace distribution. One additional detail, we add additional gamma parameter, which can adjust the weight of the log prior term, which we choose using a cross-validation procedure. This makes our algorithm more robust against any aspect of miscollaboration in our prior. Okay, with our reconstruction framework in place, we can now start to do analysis of how variations in early vision influence image reconstruction. For all our analysis below, we use images of a resolution 128 by 3, and a 1-degree foveal mosaic, which consists about 10,000 cones. One interesting variation we're going to look at is the number or proportion of different cone types. Here, I'm showing you cone mosaic of 8 human subjects measured using adaptive optics techniques. As you can see, across different individuals, there's a large variation on the proportion or number of L and M cone, or the L-M cone ratio. On the contrary, the proportion of S cone stays relatively stable and small. To understand the implication of such variation, we calculated the average root mean square reconstruction error across a dataset of natural image and how it changes as a function of proportion L cone or L-M cone ratio. As you can see, a large variation in the L-M cone ratio, approximately tenfold in either direction, has very little effect on image reconstruction, presumably due to the fact that the spectral sensitivity of L and M cone largely overlaps, and there's high degree of correlation among spectral channels. On the contrary, if we repeat the same analysis with proportion S cone, we see a clear optimal around a small S cone proportion, around 10%. This is likely due to the additional factor of chromatic aberration for short wavelengths. Our result is consistent with previous attempts using simplified model of retinal mosaic and information theoretical measurements. Together, we suggest that the variation of L and M cone and the stable proportion of S cone can both be explained by retinal mosaic design that aims to maximize the amount of information transmission given the statistical regularities of the environment. To further validate our conclusion, we repeat the same analysis of varying the L-M cone ratio on synthetic image dataset that are Gaussian distributed, for which we have explicit control over spatial correlation or correlations of pixels in adjacent spatial location, and chromatic correlation or correlation among the R, G, and B channels. As you can see, for high degree of spatial and chromatic correlation, similar to natural image, we obtain very similar pattern. However, for much lower spatial and chromatic correlation, we obtain a much higher dependency of the average reconstruction error on the L cone proportion. And you can also see a rather clear optimal around the even split of L and M cone. Now I want to draw your attention to the extreme variation of L-M cone proportion or dichromacy. As you can see, we obtain large reconstruction error, presumably due to the loss of color information. However, our methods also allow us to directly visualize this kind of information loss. So here I'm showing you example images reconstructed from dichromatic mosaic. For example, protanopia, meaning L cone missing, deuteranopia, meaning M cone missing, and tritanopia, meaning S cone missing. Interestingly, the visualization generated by our algorithm is qualitatively quite similar to results produced by other algorithm that is explicitly designed to predict the visual experience of dichromacy by explicitly including psychophysical measurement, despite the fact that our algorithm is operating under completely different principles. To summarize, we build an image computable model of early vision and use ideal observer analysis with Bayesian image reconstruction. This allows us to analyze variation of cone proportion as optimal design of retinal mosaic that aims to maximize information transmission. And our method also provides us with a direct visualization of information loss, such in the case of dichromacy. Thank you very much for your attention.